is a relatively new way to make precise changes to the DNA in cells and organisms.
That's what we've been doing. We just know how to do it better today than ever before.
For the first time, scientists have used the gene editing technology known as CRISPR inside a patient.
To cure diseases in people today, things where people really need that help.
Are we wise enough to tame ourselves in the presence of this awesome power that previously was only in the hands of nature?
You wake up in the morning, drink your coffee, get ready to attend your lecture. It's a medical lecture on respiratory diseases. You try your best to focus. You pick up the first couple of words in the lecture, then fade off. Cystic fibrosis is a hereditary disease that affects the lungs and digestive system. Although you didn't pay much attention, these words stick in your head, maybe because one of your relatives or someone you know has the disease, and this reminds you of how unfortunate it is to be born with such condition. Humans, to put it simply, are prone to unsatisfactory things. We can be short, we can be tall, we can be ill, we can be depressed. We can have cells that spaz out and fill our bodies, and the list goes on and on. What if we had the power to control all these negative outcomes? We learned that a code in our body determines everything about us. But what if we could edit that code, remove parts of it, add new stuff to it, fix it? Nothing would stand in our way, or would it? Our DNA consists of 3.2 billion letters. Letters that are in a specific sequence. A, T, C, and G, repeated, interspersed, and sequenced into a strand. Every sequence of 3 consecutive letters is called a codon. Every codon is for a specific amino acid. Amino acids bind together to create proteins. Each protein carries out numerous vital functions in our body. Each sequence has a function, to the point that a change in just one letter of the sequence can create various pathologies. Sickle cell disease is a group of blood disorders typically inherited from a person's parents. Note the word inherited. Since the disease is passed through the generation, it is definitely a genetic disease. It is caused by a single change in the sequence of DNA that is responsible for the synthesis of beta hemoglobin. Hemoglobin is a protein in your red blood cells that carries oxygen to your body's organs and tissues. Beta hemoglobin is a component of hemoglobin. 2 alpha hemoglobins bind together with 2 beta hemoglobins through magical bonds and form the bigger and better protein, hemoglobin. Beta hemoglobin contains glutamic acid, one amino acid in its structure followed by various other amino acids. The codon which corresponds to glutamic acid is GAG. When the A in GAG is substituted with a U, becoming GUG, the glutamic acid is exchanged with another amino acid called valine. Now valine is in the structure of beta hemoglobin instead of glutamic acid. A simple change in the sequence. It wouldn't harm anyone, right? Wrong. You see, the shape of a protein defines its structure. Every protein has a unique 3D structure. Change the structure and the function of the protein becomes compromised. The valine will cause the beta hemoglobin to fold in a completely different way. And since beta hemoglobin is one of the backbones of hemoglobin, the whole structure of hemoglobin is changed, comprising its function. All that due to a simple change in one letter buried between another 3.2 billion letters. Cystic fibrosis, thalassemia, Marfan syndrome, Huntington's disease, cancers are just some of the diseases caused due to genetic mutations. Let's take a step back. In 1987, Yoshizumo Ishino, let's call him Yoshi, and colleagues at Osaka University in Japan published the sequence of a gene called IAP belonging to the gut microbe E. coli to better understand how the gene worked. The scientists also sequenced some of the DNA surrounding it. They hoped to find spots where proteins landed, turning IAP on and off. But instead of a switch, the scientists found something incomprehensible. Near the IAP gene lay 5 identical segments of DNA, which were composed of the same repeated sequence. At the time, this peculiar finding was not well understood, and when the scientists published their results, they could only write, "The biological significance of these sequences is not known." Fast forward to 2002, the same sequence was found in a staggering number of species of microbes. Although it was not well understood what the function of the sequence was, but it was given the name Clustered Regularly Interspaced Short Palindromic Repeats, or CRISPR for short, due to its prevalence. 3 years later, 3 teams of scientists independently noticed something odd about CRISPR spacers. They looked a lot like the DNA of viruses. And then the whole thing clicked. At the time, Koonin, an evolutionary biologist at the National Center for Biotechnology Information in Bethesda, MD, had been puzzling over CRISPR and Cas genes for a few years. As soon as he learned of the discovery of bits of virus DNA in CRISPR spacers, he realized that microbes were using CRISPR as a weapon against viruses. Then, a bunch of yogurt makers came to a conclusion that CRISPR is used by bacteria to splice genetic sequences from viruses, granting it immunity from that virus. By 2012, CRISPR was still being studied by scientists, along with its lifelong friend, Cas. The CRISPR-Cas9 sequence would program enzymes to splice exact points of the DNA and replace them with other sequences. Since then, and to this current day, many experiments were made testing the capability of this finding. Rice was given immunity against invaders. Dogs were made into buffed dogs. Pigs were made into small pets. Elephants were made into hairy beasts. And the list goes on and on. Scientists would come to realize the amount of power they had captured in their grasps. There was no downside. This piece of technology can eradicate all genetic-related disorders. It is a leap in biological research. So, CRISPR is a cool tool and all. It can do great stuff. But why haven't we heard about it being used in humans yet? When scientists tried to use this technology in human embryos, in an attempt to stop genetic diseases from developing, they were faced with various backlash, simply because it was unethical to do so. This is because there is a chance that the procedure will work, and the baby will be born free of the suspected disease found in his genome, or that CRISPR will not be as precise and splice other parts of DNA not related to the mutated sequence, causing unintentional diseases like cancers. And now the baby carries genetic factors for cancer that will be passed down with the generation to come. To put it in simple words, CRISPR is still being studied. Despite that, in 2018, Chinese researcher He Jiang, where do they get these names from? Let's call him He. He announced that he had edited 2 human embryos to attempt to disable the gene CCR5.
We're also sending a little bit of protein and instruction for gene surgery,
which codes for a receptor that HIV uses to enter cells. He said that twin girls, Lulu and Nana, had been born a few weeks earlier. He said that the girls still carried functional copies of CCR5 along with the disabled CCR5 and were still vulnerable to HIV. The work was widely condemned as unethical, dangerous, and premature.
I understand my work will be controversial, but I believe family need this technology, and I'm willing to take the criticism for them.
Other than being used in embryos, the CRISPR mechanism was used in an attempt to treat lung cancer. Since cancer cells were growing immune to chemotherapy, immune cells were taken from the patient, and drug-resistant DNA was forced into immune cells, teaching them to attack that sequence of DNA wherever they find it. This did prove efficient as patients didn't have any other side effects. Until now, CRISPR usage to treat cancer is still being studied immensely, hoping that in the future, cancer will be an easy enemy to fight off. In 2019, CRISPR gene editing therapy was used for the first time to treat sickle cell disease. the disease we mentioned at the beginning of this video. CRISPR Therapeutics and Vertex Pharmaceuticals Incorporated reported that the US Food and Drug Administration had fast-tracked CTX001, an investigational gene-edited stem cell therapy to treat patients with sickle cell disease. CTX001 gives the patient the ability to produce fetal hemoglobin, a type of hemoglobin found in the fetus, which does its job better than the awkwardly bent hemoglobin. Victoria Gray is the first patient to receive this kind of therapy. Preliminary data have shown that the CRISPR gene therapy is working, but it will take more research, a larger cohort of patients, and monitoring over years to assess the long-term effects of this genetic therapy. But what is the difference between experiments on adults from experiments on the embryo? Aren't they both the same genetic changes or whatever? Well, genetic changes made in the embryo will be passed to future generations, while genetic changes made to mature cells in the body, like immune cells for example, will be confined to that individual, making the whatever side effect that occurs in that person die with him. We can teach our immune system to fight off organisms it does not yet understand. We can teach our cells to rebuild itself in a proper way to do its function properly. If you go on Google right now and search for smallpox, it will read, "Smallpox was an infectious disease caused by one of two virus variants, blah blah blah." Was. We have already eliminated a number of diseases, and perhaps we can do more. Perhaps one day you will wake up, drink your cup of coffee, and search for cystic fibrosis, and read, "Cystic fibrosis was a hereditary disease that affects the lungs and digestive system." Or look for thalassemia and read, "Thalassemia was a blood disorder passed down through families." Or search for malaria and read, "Malaria was a life-threatening disease." Perhaps we will never be able to bend nature, and we will never understand gene editing fully. In either case, we are just humans. Apart from treating diseases, one cannot ignore the fact that it is urging to create superhumans. That is, humans with more muscle mass, better eyesight, better food digestion. These thoughts can only traumatize scientists. It is the thought that because of we can create humans that will overcome us, a new era of humans that are able to do everything better than us. Machines, but with feelings. Thanks for watching.